Most of you know Carson, but Carson Coriell. He's going to be presenting his master's work to this point. So for those of you who didn't know this, Carson is a local yokel. He's from Anne Arundel County, Broadneck High School. I was like the river? Yeah. He came to Maryland in 2013 and was gonna do art. To study art. Then decided, no, I'm going to study biology. And then he realized all the biology majors were actually pre-med. And then he found Dr. Wendy Whittemore, who was the previous advisor in ENSP. And he switched into wildlife and got me as an instructor right away. You're so lucky.
I know.
He graduated in 16 from here with his undergrad in wildlife, and then went and worked for USDA as an LA appointment, and then he switched over to another appointment with USDA through Penn State, actually. And then I lured him back with the promise of foxes and coyotes the summer of 21, and he started his master's that fall and got no foxes or coyotes. He got COVID instead, and lots of mice. But we love Carson. He's a huge part of our lab, a very powerful personality. He drums, similar to my son. He's a big drummer. He also is a famous face at Cantlers, so we hear about his Cantlers shenanigans all the time. And the one thing I would say before he talks to us about his work is that the project he's going to present is incredibly large in terms of people manpower and just hours in the summer. If you've trapped mice with Carson, stand up real quick, or raise your hand, And this is like maybe 1/8 of what it takes on one team during the summer. So we've had pretty massive teams that he's run. But we're really excited to have him here today to present his master's work. We did transition him to an actual full-time position. So between that on a deer project and the fact that we're waiting on analysis of his second year of data, disease data from Dr. Megan Fritz's lab in entomology, we're a little behind. We anticipate a summer finish, but we're getting there. All right, without further ado, yay.
Thank you, Dr. Mullinax, for that lovely introduction. As she said, my name is Carson Coriell. I'm happy to be here. I'm excited to present to you guys on my research. So small mammals, ticks, and tick-borne diseases all in urban parks. So we're going to start. Quick outline of what we're going to run through. I'll do a brief history about what Lyme disease is, some of the objectives of our lab and of my study, some of the methods we used over the last couple of years to get through all of these kinds of objectives, some of the modeling that we plan to do, and some of the plans that we have for how that modeling will affect park managers in the future. And I'll touch a little bit onto some of the ongoing analysis between modeling and disease. So essentially this is going to run like every seminar presentation you've ever seen. It's going to have a very similar view here. So before we get started, quick show of hands, how many people in the room either know someone or they themselves have Lyme disease or been infected with Lyme disease? High hands, high hands. Okay, so that's a lot of people. So now keep your hand up if you know someone, the person you're thinking of is maybe not like considered an outdoorsy person. They don't hike and camp a lot or spend a lot of time in forest. They're just an average person, right? That's still like half the room. That's a significant amount. Come on in. That's a significant amount of people that are familiar with Lyme disease. So this is a real problem, right? So somebody should do something about this. So that's kind of what our lab thought as well. So we sort of set out to do that as well, but I'm gonna hide these controls. You'll see me do this often, I imagine. So quick background about Lyme disease. As you saw from the show of hands, it's one of the most common vector-borne diseases in the United States. There's estimates right now that it's around 30,000 cases per year in the United States, but because of the way that they confirm these estimates, it has to be through clinical infection, like rashes and things like that. So the true numbers the CDC reports is actually probably closer to 300,000 per year. So it's a huge problem that we're really trying to figure, that we're only grasping at how to solve and trying to figure out what we can do to mitigate these. So the primary spreader of this is the deer tick, Ixodes scapularis. I have the life stage over here on the left. Many of us who worked in the tick world have seen this many, many times, so you'd be familiar with it, but basically it's a 3-year life stage. The ticks are born as larval. In a larval stage, they get onto a host species, then they will fall off into the leaf litter, molt, and then go into the nymphal stage, and so on and so forth, all the way up to the adults, where they will then attach onto a white-tailed deer, which is where they get their name, the deer tick. I'll probably refer to them as Ixodes scapularis from this point moving forward. But in those early stages, that larval and that nymphal stage is really when it's important for the small mammal. The small mammals play an important host species for them. So they're a blood meal for them, but that's where it's possible they're getting the infection from. And I'll touch on that in a moment here. So, there we go. A little bit of history about Lyme disease, where the name comes from. You might have heard that they were made in a lab in Connecticut. That's not true. They are originally derived from the town of Lyme, Connecticut, because that's where it was first discovered. There was a lot of young children, children and young adults that were contracting rheumatoid arthritis-like symptoms. So a lot of joint pain and rashes and headaches and things like that. And we now know today that it's not likely that children are getting arthritis, but it was actually Lyme disease. But at the time, we didn't really have this information. So it was just kind of what's going on, big mystery at the time. And it wasn't until the early 1980s that this guy, Dr. Berdorfer, discovered the spirochete within the gut of the deer tick that is responsible for the spread of Lyme disease. So it's the bacteria. It's called Borrelia burgdorferi because, like every great scientist, he named it after himself. So now we have to go through and say his name every time, but it makes it easy to remember who he is because it's named after him. So 1970s, we have this issue. Early 1980s, we finally figure out what it is. And then from there, we can pinpoint our enemy. So I share this slide for two reasons. One, it's kind of fun to see everybody squirm a little bit when you put an arachnid on the stage or on display here. It makes people uncomfortable. But two, to just really get at the anatomy of these ticks. So what you guys might consider to be the head of the tick is actually just its mouth parts. They don't actually have a head. They don't have eyes. Some species of ticks do, but this one does not. So just kind of knowing, who we're up against here. The larval stage, obviously, nymph, adults, as we mentioned, but for the purposes of my study, it's going to be a lot of the larval and nymphal ticks that we're going to be looking at. Here's the reported distribution of the ticks throughout the years. So starting in 1996, you can see that it's a little sporadic, but as we move forward to today, it's blowing up all over the landscape. We're really getting these ticks, all over the place. And we give these kinds of talks at conferences and things like this out west in, Seattle and places like that. And they're like, oh, Lyme disease, we don't have that problem. You will, it's coming. So the distribution is getting larger and larger, and we're trying to figure out, some mitigation techniques of what we can do to prevent this. So first we have to understand the problem. So with that, we're going to, just a brief synopsis of how somebody might contract Lyme disease. The tick does what we call a questing behavior, where they take their two front legs, stand on the edge of vegetation, and kind of sway their arms back and forth and hope to grab onto something, presumably a human at some point, right? And so it burrows into your skin. After it's on you for a select number of hours, depending on the disease. For Lyme, it's 20 to 24 hours, or 24 to 48 hours. That's when you start to contract all of these symptoms. So if you catch it early, you can get medicated by doxycycline, and there's lots of things we can do. But if you're unaware, most people think, I wasn't camping this weekend, it couldn't be that. Then you can actually get a lot of issues with this if you don't catch it early. So why are we focusing on small mammals? As I mentioned earlier, they are an important host species for the ticks. That's where they're getting their early stage blood meals. But it's also the disease reservoir. So ticks are actually born sterile for Borrelia in their early stages, and then they get onto a small mammal, specifically a white-footed mouse, and that's where they're contracting the disease. Enough blood meals coming from these white-footed mice, it's going into the tick, and then throughout the life stage, it's carrying it on and potentially getting onto a human at a later life stage down the line. Some of the other instrumental players here are chipmunks, shrews, and squirrels. You'll notice that I have yellowed out gray squirrels, and maybe if somebody has a good memory, ask me a question at the end why I have done that, and we can get into it, but it's kind of a whole other thing I tried to solve, but it's going to be, anyway, ask me again at the end if you're interested about gray squirrels. This is a quote from Dr. Asfeld, who is the sort of leader in small mammal research over the last 20 years, and his quote is, "Where the mice go, so too go the infected ticks." Basically just saying that if you have a lot of mice, you're bound to have some infected ticks in that area as well. And he's kind of the front runner for this research. A couple examples of what we're looking at in the field is, this is an ear of a mouse. And there's obviously a tick on there. So these are really small in some instances. People tell me all the time, like, oh, I had a tick on me. And I say, oh, how big was it? And they say, well, it was really small. I'm talking really small for some of these larval ticks. And this is a good example of just one, but some of the individuals can be heavily inundated with ticks. I mean, it's not uncommon to find a small mammal that has lots of ticks on them. And if you imagine that these ticks were sterile and this mouse had the Borrelia in it, now all of a sudden all of these ticks are potential disease reservoirs. So this is something that could be a serious issue very quickly. Specifically honing in on Maryland as a state, because obviously University of Maryland is what we care about. These are the estimate number of Lyme disease cases over the last 20 years or so. Again, it's the confirmed cases. So this is 31,000 confirmed cases, but the true number reported by the CDC is actually likely closer to 300,000 just in these last 20 years in Maryland alone. What we're going to be focusing on, and I'll get to in the next slide, is Montgomery County. So that's this really deep red area here. Obviously, we have a lot of ticks there. It's a lot of, very dense urban areas, as most of you are familiar with the area if you grew up around here. I lost my thing. I can't get rid of my floating meeting controls. There we go. So here is reports of Lyme disease within Montgomery County. You can see that there is a little bit of fluctuation. I mean, ticks are, they are very sensitive. So there's not always the same population from year to year, but we are trending upwards to have more and more ticks on the landscape in Montgomery County. I believe the estimates here, do I have a good number for it on the screen? No, but you can just kind of see that things are moving upwards and we're getting more and more ticks in Montgomery County. So to take a step back first, before we get into my specific objectives, just our lab, the Applied Spatial Wildlife Ecology Lab as a whole, is really interested in figuring out the disease metrics of all the animals in the landscape. So we have a huge camera trapping array of about 100 cameras put out over the last 2.5 years. Right now, we're somewhere between 900,000 and 1 million images that are currently being processed that are going to get at the density of all kinds of animals all over the landscape. Everything from the deer, which takes up most of the pictures. Obviously, we have a huge deer problem here, but really anything, raccoons and anything that we can get in front of these cameras. So you might be asking, and I'm sorry, this is a huge project led by PhD candidate Grace Hummel. She can be seen cackling in the middle of the room. So my project is going to kind of feed, thank you, Grace, feeding into this larger project set by the lab. So you might be asking yourself, if you already have this huge camera study and you're already taking 900,000 images, then why would you even bother going out to trap the small mammals? Can't you just get the information from the cameras? And the true answer is that photos like this very rarely happen. And if you're in the back and you can't see, there's a little mouse on the end of the stick here. So it's not impossible to get small mammal density estimates from camera traps, but it would be a different survey. It would kind of just be a whole thing. And there's nothing like a tried and true mark recapture study, as well as obviously infection is important for our results. So we really need tissue samples from these individuals. So going out and trapping the individuals is really the best case scenario to get a whole picture of what's going on in this landscape with small mammal populations. So my own personal objectives is first to go out and find the densities of these small mammals and the ticks that are in this environment, specifically the Montgomery County, and I'll show some maps here in a moment, but that's kind of the first step, right? We have to figure out what numbers of these animals are in here before we can do any sort of modeling after that. So the second objective down the line will be to create a rapid assessment for park managers who are able to walk into a park, assess certain types of vegetation, invasives, things like that, and be able to get a quick estimate on what our likely small mammal populations and tick populations might be. And they can use that for hopefully to be able to mitigate disease by, implementing IPM strategies and things like that to lower tick numbers and hopefully lower infection rates as well. So some of the methods that we set out to accomplish these goals were to pick a few parks that are in dense urban areas, because while this small mammal population study, small mammal Lyme disease study is not new, in urban environments such as ours, it is very rarely done. So we're sort of novel in the aspect that we're choosing to do this in very heavily urbanized areas. So we chose 2 parks, the long and stringy linear park is Rock Creek Park, if you're familiar, and the more open space park is Cabin John. I apologize if you're red-green colorblind. I am aware that this is just going to be nothing for you, so just bear with me here. Each site was selected with purpose, knowing that there was a lot of human activity. There's campgrounds, there's maintenance yards, there are in between shopping centers and things like that. So these are really kind of next to a lot of human use activities, and every single site is heavily inundated with active trails of people who walk their dogs and hike through them daily. We also went out and did a pilot study to make sure that there were ticks at all of these sites before we did any sort of sampling to make sure that this was actually going to work. So the next slide here is just a kind of a zoom up. So the photo on, I guess, your left is a zoom in of one of my sites here in Rock Creek. And you can see that it's kind of sandwiched between a neighborhood, suburban area there, and then sort of like a shopping mall center here. So it is still very, visibly, even from a backup map like this, that there are a lot of like human activity in these areas. And from a GIS point of view, we can look at these layers and their landscape covariates to figure out, this is impervious surface, this is forest, successional habitat, things like that. So more on this stuff later, but just so you can kind of see what we originally picked and what it looks like in a GIS layer. To zoom in a little bit further, you can see each transect has 5 lines of 10 traps each. So that's 50 traps there. This is what they look like. Straight lines don't exist. We actually choose small mammal habitat over top of just making it a straight line. You want to make sure that there's actually mice in those areas. And then just a cute mouse for the picture. So to kind of sum up all of the trapping out array here is that there are 2 parks with 3 trapping arrays each. So that's 6 total sites with 50 traps in each site. So that's 300 total trap locations. In order to get density estimates, you have to trap each one of these locations 3 nights in a row, and that would be for each month of the summer, so June, July, and August. So we did tons of extensive trapping, so it's 9 total trap nights per site per year. So multiply that if you want, but it's just, take my word for it, it's a lot of trapping efforts here. What this actually looks like on the day-to-day is that the folks here seated here in the front couple rows would meet me in the lab at 5 in the morning, which often means waking up at 4 o'clock in the morning. We'll gather all of our gear, drive out to the field site, collect all of the traps that we would have set the night before, and go through and process all of the mouse that we have, or whatever happens to be in the traps themselves, taking tissue samples and things like that. Here you can see me attempting to explain while people either pay attention or don't. But sliding through to what we actually did when we would get an individual is we would take the individual and weigh it. We'd give it a unique ear tag with a color and a number, so I would be able to know exactly who this individual was when I recaptured it. Take tissue samples for disease testing. Any ticks that were on the individual, we would pull those off for testing as well. And biological measurements, sex, weight, is it pregnant, how long were its tails, a lot of those morphological data points as well. So we collected pretty much anything we could on these small mammals while we had them in hands. And then everything was, don't restart the computer, everything that we collected was then put back and safe. This is a live capture study. We did not euthanize any of the animals. They were all put back exactly in the same place they were picked up. So just so that anybody might be worried that we were injuring the animals in any way, that is not the case. So this is what we want, right? We're aiming specifically those Paramyscus leucopus, the white-footed mouse, that's our target species. But when you put a box full of peanut butter out into the woods, you get a lot of crazy things that start interfering with them. Specifically, a lot of raccoons like to mess with them, turtles, we've caught wrens and things like that. So that's what we want, and we just kind of deal with the other stuff as it comes. In addition to the small mammal trapping that we did, we're also interested in the questing ticks that are on there. So just ticks that are free on the landscape, not currently attached to any host species. So what we did for this was we have a 200-meter radius circle that we put in the middle of these trapping transects, and we would go through and do tick dragging. And that's what you can see Grace here doing in this photo, essentially a slow stroll through the woods carrying like a denim sheet behind you. You stop every 10 meters or so and pick up any ticks that were on there and categorize them, GPS them, and things like that. We would typically sample between March and November on optimal weather days, so ticks like it hot, humid, sunny, any of those kinds of days where we were able to go out and do some tick sampling, we would try to get out there in addition to the other things that we were doing. The trapping grids themselves were assessed for whether or not they were quality tick habitat. Was it too wet? Was there an open canopy? Things like that. So I'll get into some of that during my results, but just know that, some of these areas are higher potential for ticks just based on the landscape that they're found in. And here's just some photos about how we do this, so you walk through, you pull ticks off of you, and sometimes we call them larval bombs. Sometimes you can have as many as like 200 ticks crawling on your sheet, on yourself. So it's really not for the faint of heart. That's kind of the first day as an intern. We take you tick sampling, and if you come back the next day, you can stay. So if you're okay with being out in the hot sun and getting covered in ticks, then you're welcome in our lab. The last thing that we did here in terms of getting at my objectives was vegetation sampling. You can see that it kind of wears on you after a while. We've done a few of these. So you saw the robust GIS layer that we had, but there's nothing like boots on the ground for some of this stuff. So we went out, we did 1 meter square transects on each 5 times within each study site, where we would assess canopy cover, the relative species, leaf litter, down woody debris, things like that, things that make good habitat for small mammals and ticks. And then we would do this multiple times. So for my study, you can see that we did this, I guess it's around 30 times or so, but remember that my project is a subset of a much larger project. So I think in total we did around 150 of these over the course of one summer. And we're not done. We're going to keep going throughout the next year as well. So some of the estimates that we're going to use in the modeling to figure out how to actually get at these density estimates was using Seeker. It's spatial explicit capture recapture, and basically what this does is it takes the number of individuals that you have in the landscape, the traps that you've sent, or the traps that you put out into the landscape, and then the occasions, which is just a successful trap. That means that you either 0 or 1, you've got an animal or you did not. And then from this, you're able to derive density estimates based on how many individuals you had marked and how many individuals were unmarked, and how many recaptures you had with that. Another assumption that comes along with this is homogeneous point process, which just means that every single trap is, you're not less likely to get a trap or a mouse at 1A than you are at 1B. They all just have equal chance probability of getting an animal in them. And as well as homogeneous catchability, which means that the ear tag is not limiting the amount that we were going to catch. You still catch them with ear tags or not. It doesn't interfere with their ability to be catched. And they're really minimally invasive, so we don't believe that that would have any instances there. And then just the activity center is where you can kind of get at a estimated home range or a nest site of these individuals. So that's kind of what this little chart here is showing. So what I did was use program density, which is really just an interface for using R. Using that Seeker package, we did closed population models with maximum likelihood estimations across a Poisson distribution. And so what this really kind of is telling us is how many animals we captured versus how many animals we recaptured, and trying to use these sort of estimators to find density estimates. And some results. So what did we actually do? We set out to get all that questing tick information and vegetation estimates, but we are not done processing all of that. So I'm going to kind of gloss over it here and dive right into the small mammal stuff next. And here's just kind of some raw data about what we actually collected. So obviously Paramiscus, the white-footed mice, is the most heavily trapped animal. It's what we were targeting, but we also got chipmunks, that is gray squirrels, some Carolina wrens, some shrews, flying squirrels, a sex ratio just to show you kind of the percentages of what we had and the number of tissue samples that we collected. And this is the number of host ticks that we pulled off of those individuals as well. And then here, it's a little bit of a busy graph, and I'll simplify it in the next slide, or table, I'm sorry. But what's important to note here is that the number of captures is obviously much higher than the number of individuals. And again, that goes back to being able to run spatial explicit capture recapture, is you have a real estimate of the numbers on the landscape by trapping, repeat trapping efforts every single day. And we kind of can account for that. There's something known as trap happy versus trap shy. Some animals are like, free anesthesia and peanut butter, I'm in, I'll be here every day. Whereas some other animals are like, I'm never coming back to that. So you can kind of account for all of this through basic modeling and equations. But really what we're focused on here is these density estimates. So remember that we did 2 unique parks. So the first 3 is that Cabin John site, that was that big open park, and the Rock Creek site was that small, more linear space limited site. And we're seeing much higher density estimates in that limited space site than we are the big open one. So there's a lot of potentials for this. It's possible that the animals just don't have as many other places to go, so they're kind of competing for space here. So lots of kind of hypotheticals that we could get at here, but this is just basically showing
What's the unit?
Of the density estimates. These are just individuals.
Per area?
Oh yeah, per 1 hectare, sorry, yeah. So each small mammal trapping grid was 100 meters by 100 meters, so 1 hectare across. And this is just an easier visual to kind of get that same numbers. It's really easy to look at this and see that Cabin John had much lower density estimates than Rock Creek did. And obviously there's differences from year to year. There's a lot of variables that come into why you might get different numbers each year, but it's pretty consistently showing us that Rock Creek is turning out higher density numbers than Cabin John. And here's an output from that program density where we can see the home ranges, or I'm sorry, the trapped individuals that we had. So it's a little busy. We don't have to spend too much time on this, but the checks are the actual traps, and then the colored dots in the lines are the individual that we trapped and where they moved. So you can see just at a glance that Rock Creek has more of them, and they're moving a lot more across the landscape. They're kind of competing for these traps, whereas some of the other sites are able to, this is my nest spot, nobody else come over here, you know? So just at a quick glance here, you can see that Rock Creek is showing a lot more movement between the traps and a lot higher density estimates here. And some infection data. So remember, we took those tissue samples and we had those tested for the presence of Borrelia and many other diseases, Rocky Mountain spotted fever, anaplasmosis, rickettsia, just to name a couple of them. So we showed actually a 29% infection rate, which is pretty standard across what literature shows us, which was pretty unexpected because if you remember back to that early map I showed of Montgomery County, we had a lot of heavily reported cases of Lyme disease. So we would have expected to see a lot more cases of infection, but that's just not the case of what we're seeing here. And I'll break this down a little further in the next couple slides. But the other graph is just showing some infection of the percentage of infected tissues, what species did they belong to. So arguably, or probably obviously would be white-footed mice had the most of the infection, but also chipmunk and squirrel showed up for infected tissue as well. So here's the infection data by park. So 61% of the samples at Cabin John tested positive. So meanwhile, 7% of the Rock Creek samples tested positive. So we had like, I think 161 samples at Rock Creek that we tested. And I think it was something like 11, I don't remember off the top of my head, but only 7% of them that are showing up. So this is pretty surprising from a data analysis perspective. And then to break them down by site. So there's 3 sites in each one. Again, Cabin John showing the most, but 50% of Cabin John 1 was showing positive, 69% of Cabin John 2, so on and so forth. Noticeably absent from this is Rock Creek 2 and 3. We did not have any positive tissue samples from those sites, which we were actually all pretty surprised about as well. So what does all of this mean, right? Oh, well, actually, I'll get into what it all means, but the ticks that we pulled off of the mice, the host species as well, we tested those for infection. So the 2022 data, we got very low infection rates. Only 1 positive for Borrelia, and it was actually a co-infection with anaplasmosis. And while that may seem surprising from a just quick glance, it's really not that surprising when you remember that the ticks themselves were born sterile for Lyme, then they would have contracted it from the small mammals that they got onto. So if we are pulling ticks off of possibly before they were able to get their blood meal and get infected with the Borrelia burgdorferi. So that's 2022. And here in 2023, we actually saw a little bit more infection and new types of infection. So anaplasmosis is now in green there, and Borrelia burgdorferi is in red. But we also saw some Babesia microtei as well, which if you're familiar with Babesia, it's sort of a malaria-like disease, which causes fever and headaches and things. So it's just another bad thing to get from ticks. You kind of want to be careful when you have any ticks on you in general. But this is some of the infected breakdown that we had from those host species ticks. As well as the free questing ticks that we went through. Now, I don't have any data to show their infection, but these are just the counts that we had from them. Again, Cabin John is showing lots of ticks where Rock Creek was not showing very many. And you may think that these numbers are a little low because I said earlier in the presentation that you were constantly covered in hundreds of ticks, and that's because those are larval ticks. So these graph is only showing nymphal and adult stage ticks. We're still counting the larval ticks. It's a lot of them. So we're gonna get some of those estimates here in the future, and I'll be able to check back. And then the ticks on the bottom are just showing what the primary species was that we found. So in these pictures here are the Ixodes scapularis, and the other one is Amblyoma americana, or the Lone Star tick, if you're familiar. So a lot of these different ticks are associated with a lot of different diseases, and obviously for my study, we're mostly concerned about the Borrelia, but this does just kind of show us that there are other ticks on the landscape and things to be wary of as well. And what we found was a huge surprise is not only are we having the deer tick and the amblyoma that we would have expected, but this guy, the Hemophasophilus longicornis, the Asian longhorn tick, showed up on the landscape as well. And this is actually really scary for people because they are parthenogenic, which means they can clone themselves. So they're all female right now in the United States, but that means that one female can lay 2,000 eggs. All 2,000 of those ticks will be guaranteed female and can lay another 2,000. So this exponentially is exploding over the horizon right now. And one of our sites specifically, one of our Cabin John sites is just completely inundated with these ticks. So we're kind of seeing I think we were one of the first people in Maryland to find these, and now we're having no problem finding them. It's really one summer you find a few, the next summer it's all that you can find. So this is just kind of a warning label that these things are out here. We're still studying the implications of what this means for the landscape and disease potential, but it is scary, I think. So some of the synthesis, what does all of this mean, right? So what we're seeing is that Rock Creek specifically has high levels of mouse density. We caught a lot of individuals there. We know that there's a high level of density, but really low infected tick numbers. So we're getting, the potential there is that it's just not quality tick habitat. It's wet, it's closed canopy, it's kind of, we call it like a swamp, but it's just sort of a really marshy area compared to the Cabin John. So there's low levels of disease there, even though we have lots of ticks. Or I'm sorry, lots of mice. The other side of this would be Cabin John, where we're having lower levels of mouse density, but higher levels of tick numbers, and we're getting high prevalence of disease. So contrary to what Osvald said, that quote I shared where mice go, so to the infected ticks, what we're actually seeing is it might not be as important how many mice are in the area, but the tick habitat, whether or not this quality tick habitat is going to relate to infection on these landscapes. So really trying to still piece this together, but this is just a preliminary kind of show at what we're seeing here is that low levels of disease correlate to the poor tick habitat, and having a lot of small mammals on the horizon might not actually be a correlation to high levels of disease. So some of the next steps, obviously, we mentioned that I'm currently staff working on another project, so I am a little behind on some of this research, but some of the things we want to do next is actually determine whether or not these density estimates are accurate. So we will run them again and bootstrap them to find confidence intervals to see if we can actually ensure that what we've done is the correct way and the accurate way to do it. And we will, right now the density estimates that I've ran has been one session at a time, meaning one summer I ran the density estimates, the next year I ran those density estimates and just compared. But what we'd like to do next is run them as a continuous one session to find out density estimates for the entire project. And then we'll compare these results. So this is all spatial explicit capture recapture, but we'd like to also compare them to Cormac Jolly Sieber, which is a very traditional method of doing this, as well as a robust design. So these are kind of the next steps on how to assure that these density metrics are accurate and the best reported that we can do. What we'll also be doing is using some of the covariates in the landscape to determine whether or not these are making a huge impact. So some land cover types, successional habitat, density of trails, roads, impervious surfaces, things like that. So we're able to take those metrics into GIS and R and figure out whether or not the proximity of these human landscapes and these plant species and things like that are having a big impact on the ticks and the mice that we're finding in these areas. And then from there, we'll be able to do some generalized linear mixed models using random site variables to figure out, again, tick density is going to be important and mouse density, but really the end goal here is trying to figure out infection rates, right? So the Y equals will be essentially the disease prevalence, and then the tick density and mouse density will feed into that. So that's kind of coming up next. So if any of this interests you, I encourage you to check back in with me for my thesis defense this summer. So it is still a lot of work to be done. We've done all the fieldwork aspects of it, but we still obviously have a lot of modeling to do and a lot of statistics to run. And quickly before I move on, most people get to this acknowledgement slide and they say, I'd like to thank my committee members and my lab mates, and then they move on. But this is not the case for me. I love these people. They have worked so hard for me. They woke up at 4 a.m. every day, all summer, came out, did all kinds of trapping in the hot sun. I'm the one standing up here presenting this, but I feel like I'm standing on the shoulders of giants. These folks are not just interns and technicians and lab mates, they're my friends. They've become just such a huge integral part of my life and my lab and the research that I've done. So I really wouldn't be here without these guys here. So I really want to thank you all for doing that, as well as thanking my committee members. A special shout out to Dr. Megan Fritz and her graduate student Thea Bliss for their disease testing for us. As well as everyone else, the University of Maryland as a whole, Montgomery Community, or the Montgomery Parks and Recreation for allowing us to do this study. So really everyone that's been involved with this is really, we wouldn't be here today if it wasn't for everyone's hard work and dedication. And with that, I'd be happy to take some questions. We'll go in the back first.
Hi there. I have two questions. One, what's up with the eastern white squirrel or gray squirrel?
Mm-hmm.
And then two, this is kind of adjacent to your project and also a question for you. And Jen. What do you attribute your very cohesive lab dynamic to? Because honestly, I'm a bit jealous because my lab is as tight knit, I think, and really just well put together. So what goes into
Okay,
all that?
a couple good questions. And for those that are just kind of like wanted to look at more pretty pictures and stuff, here's a little post-credit scene of you can read of some tick myths that I'll debunk while I answer those questions. So the first one was the small mammal, the gray squirrel, right? Why did I yellow that one out? So squirrels, it's really kind of contested still in literature whether or not squirrels are a dilution host, meaning that they're not holding the reservoir for the disease. They're just getting ticks on them. The ticks are taking the blood meal, not getting the disease, and then moving on. So in order to get at that answer, my second year, I put out much larger traps, an extra 60 of them actually. So we did increased our trapping efforts by a large metric. And we just found that the mice were using them as large hotels. So we did not really increase our squirrel trapping efforts at numbers, but we got a lot of mice that were a lot happier to be in much bigger protected areas. So it's kind of we wish that this wasn't the case. We tried our best, but in order to successfully trap squirrels, it's a different timeline. Squirrels are diurnal, mice are nocturnal. I'm only one person, so really you would have to set up a different trapping array and run them at different times. So we just tried our best to do them at the same time. As to the question about why we have such a tight-knit lab dynamic, I have to just really, I don't know. We've worked with some of these folks for a long time. I really have to attribute a lot of it to Dr. Molinak. She's not just a professor to us. She's, kind of a quasi-mother. She's really kind of gotten involved in our lives in much more than an academic sense. And some of our graduate students, myself and Grace have been friends for almost 10 years. We went to undergrad together. So when we did bring in new people from the lab, it was easy to kind of just bring them into our family. So. Yeah, who else? Gabby, I saw your hand first.
Yes, thank you, Carson.
Sure.
So just remind me what's the distance between your traps?
They're 10 meters apart. So it's 1 hectare square and each trap is 10 meters apart.
10 meters.
So 100 by 100. So 10 all the way up, 10 all the way across. Yeah, sure.
I'm doing math in my mind, sorry.
No,
Keep going.
I don't know. Do you have a follow-up question with that?
Yeah, but I can.
Okay, you do the mental math, we'll circle back around. Sure, Mary.
As far as like the new Asian longhorned tick, so it seems to be like you found it at the site that had the highest tick density of the ticks that you were studying.
Correct.
So is it like, I don't know anything about tick ecology, does it seem to have like the same like habitat requirements and preferences as the ticks we already have here, so it's just gonna kind of like...
I can answer that from my own personal experience, and I would say no, but really from a literature standpoint, we're just not sure because it was first found in the United States in 2017, so it's really not like fully studied yet. I mean, it's still kind of emerging on the landscape, so people are still trying to figure out what's going on here. I will say to maybe calm some nerves in the room that currently, I don't believe that there's any disease prevalence of these ticks. So in their native home ranges of Eastern Asia and of New Zealand and Australia, they are associated with really awful tick-borne diseases like powassan. But until we start getting male ticks in the United States, then we're just getting the same parthenogenic quote. And there has been one male detected, I believe, in Ohio. So we are getting really nervous about what that means for the disease of tick-borne diseases on the landscape. Sure. Marty, go ahead.
So,
Dr. Ravenhorst.
could you talk just briefly about personal protective equipment and best practices?
Yeah, absolutely. So before you even consider personal protective equipment and all the things that we wear, you have to check yourself. Like that's the first thing we tell all of our people in our lab is I can put you in a full painter suit, tuck your boots into your socks, duct tape them, wear high socks and boots and all kinds of things, gloves, Tyvek suits and things. The ticks are crafty and they will get in anyway. So you really have to check yourself. And kind of the common phrase that we use in the lab is get a mirror, get really comfortable with yourself and look for ticks. Because as you've seen my tick myths here is that it's often associated people, you hear them say, oh, ticks are falling from the trees, check your hair. If you have ticks in your hair, that means they likely crawled up your whole body and then you found them in your hair. They're really popular spots are your waistline, places, dark, moist places that you sweat and things like that. So waistlines and things like that under your arms are really kind of like great hot spots to check for ticks. But in terms of the PPE that we use, we wear coveralls, boots, high socks, things like that. We take lint rollers out with us. So if there are ticks on us, you can lint roller them off. But really nothing beats a tick check. And it just happens to be that you have to do it yourself, you can, or a really close friend.
And then keep in mind that we can't wear bug spray.
I was gonna ask, what about?
But
Oh,
you
yeah.
should. You should if you're
Yeah.
not collecting
Yeah,
ticks.
yeah.
That is a great point.
Permethrin.
So
Yeah.
2 points on that. Permethrin is great, and
close.
DEET and things like that are effective. You'll see people use garlic sprays and scent sprays and things like that. It doesn't
None of it
work.
works.
It doesn't work. You can't use it. None of our lab is allowed to use it because we're actively searching for ticks. So if you cover yourself in permethrin and then walk through a field and go, I didn't get any ticks, there's none here. Well, it's because you just actively repelled them. So really, I think that shines again to how dedicated some of our lab techs and our interns are, that they're able to, go in and allow themselves to be crawling at ticks just for my research.
So I have a question. Would you say that your the Cabin John site is more affected by urban development than the Rock Creek site?
That's a great question. I'm not really sure. I would imagine, so just at like a kind of a glance, I would imagine that the fact that the space isn't as limited in the Cabin John site, so there's just other places that the animals can go, whereas the Rock Creek site is so limited by housing on this side and urban development on this side that they really don't have any, they're kind of limited here and stuck. So in terms of like urban development and things like that, I think it depends on what else is around the area. We did have a few instances of that where they built a road through one of my transects over the top of a year. I had one of my sites is in a power line flyway that was like really just overgrown. They only mow it once every 4 years. Surprise, they mowed it in between the years that I had started, where we reached out to them and they said that they were going to do it beforehand, but that's just the way the government works. It's a little bit slower and you happen to be doing a study involved when they do that. Yes, Grace.
It's a kind of follow-up question to your question, and I have no idea what these sites look like, but from this and from your densities, what do you think is the biggest landscape metric that you're gonna pull into your density estimate?
Ooh, that's a great question.
Mice.
I would probably suggest that edge habitat and successional habitat, these areas where things are being mowed or trails are being maintained and things like that, like we know that animals, specifically small mammals and things, love edge habitat. So really that those are the drivers for higher density and potentially higher drivers of ticks as well. So again, we'd have to run through the models to find a concrete answer for that, but that would be my just preliminary guess, would be that successional habitat and things like that. Oh, there's an online question. Go ahead, Shannon.
Hey, Carson. So great presentation, by the way.
Thank you.
I wanted to point out, I'm able to get vaccines for my dogs. So a Lyme disease vaccine for my dogs. Why can't I do that for myself?
That's a great question. So there used to be a Lyme disease vaccine and it was historically ineffective. So they kind of just got rid of it, and they're working on a new one currently. So it's just not available. It's in testing. Potentially you will be able to get a Lyme vaccine for yourself in the next coming years. But we're just not there yet. I think one of the other issues is that Borrelia doesn't really detect well in blood. So when you go to the doctor and you get a Lyme test and they draw your blood for it, it's kind of historically inaccurate. I had to do this every year to every 2 years, and the doctor always says, oh, this doesn't really do anything, which is what you love to hear from your medical professionals. But really it's like a tissue sample is really, those kinds of tests are how you are able to determine whether or not you have Lyme. Anything else? Any online folks before I continue with the folks in the room?
Yeah, that was.
I'm not a doctor, so I can't speak specifically. Yeah, maybe just something small, finger you don't like, I'm not sure. But I do know that, that's one of the reasons we didn't take blood from these animals, is that Borrelia does not show up well in the blood. So it was kind of a metric we didn't need. Although looking back, I wish that I had, because anaplasmosis does show up in blood. So that would have been a better co-infection rate if we had the forethought.
Spirals in the muscle tissue, so it gets hidden.
Yeah.
Like spirals in the muscle tissue. So it gets hidden from blood tests.
Yeah, so like your spleen is a great thing to grab. If you're not attached to your spleen, that'd be a perfect thing to test.
So I have a follow-up question to my
Sure.
previous question. So I think is it true that one of the core questions here is why? So if the disease ultimately is coming from the mice, so the ticks are getting the disease from the mice.
Yes.
And you said you had more incidence of disease, even though the mice population was smaller in the Cabin John site.
Correct.
Is that correct?
Mm-hmm.
So here's a big question. Why do you think the mice have a higher incidence of disease in the Cabin John sites?
So the question is, why is there higher amounts of disease in Cabin John compared to the Rock Creek sites? The Cabin John site is just a tick haven. I mean, there's open canopy, it's not nearly as wet, it's big humidity is a big factor with tick biology. So I think it's just perfect tick habitat. So really, you know, it comes down to what works well for the ticks, and if that is the spot that all the ticks are gonna be at, then that's where the disease is gonna kind of cycle through the system repeatedly.
But Jared, the ticks can give mice the disease also.
Okay,
So clean mice can get it from a tick.
so it just cycles between
Yeah.
the mice?
Yeah, the mice are just this kind of continuous reservoir of like holding the disease, and it's, you know, it doesn't really affect them the way that it affects us, so they can have it and not really see any detrimental effects to it, but later on down the years, when the tick that bit that mice gets on you, now all of a sudden it is your problem. Yes?
Two really short questions.
Sure.
One more question. First, when you say a Rock Creek site is wetter, does that mean, is it because of the stream overflow, or is it urban runoff?
That's a great question. So I would have to look into, I guess, the history of the park and things like that. Really, I think it's just kind of a wetland. So one of the reasons that these urban green spaces exist is because they're not good habitat for humans. You know, they're not good for construction companies. Otherwise, we'd have houses there. So because that there's this sort of wetland habitat that we can't build on, we're like, ah, cool, we'll turn it into a green space for folks, which is great for the animals and, you know, all the ecological benefits we get from urban green spaces. But in reality, that just means that it's poor habitat for ticks.
Okay. And then for your site selection, did you choose like an equal distance away from like an open space of like equal to forest or?
No, so that's another great question. So why did we choose the sites that we chose in our parks? Really, we were limited by space. We needed 1 hectare of space that we could put a trapping grid in that wasn't going to be messed with by folks that are in the parks every single day. You get a lot of people come up to you and say, well, I'll check the traps for you. I said, no, please. We do cook procedures and things like that. I can't just have you grabbing random animals here. So we were space limited in the first one. And the second one was, I mentioned we did the pilot study where we went and actually searched for ticks, and these all came back as hotspots for ticks. So knowing that there were ticks there, it was enabled to be, we had 1 hectare of space that kind of fit in with those urban landscape metrics that we were going on to. You know, there's folks here every day. So those just kind of worked out. We actually were going to include a whole other park, but the project got bigger and bigger as the days go on, and we just had to limit it to what we could be able to do in a single day.
One more quick question, because I'm like dominating these questions.
That's okay.
But do rats carry Lyme's disease? So, like,
We were hoping to get rats, honestly, especially with those bigger traps. So we know that they are, but we not to the same extent as far as my knowledge goes. But I would love to do some more literature review on specifically Norwegian rats, you know, some of the invasive species, to find out what their prevalence is as well.
And one last question for me.
It's okay.
Which site is closest to DC? Your Cabin John
Rock
site?
Creek.
Rock Creek.
Mm-hmm. Rock Creek is actually split up. There is a DC Rock Creek,
Yeah.
and then there's the Rock Creek SVU 1 or whatever it's called specifically. But so it's in Montgomery County, but Rock Creek itself extends all the way through DC. It's a really massive park. It's just so long and linear that when you look at it on a map, you think it's small, but in terms of actual like square meters of space, it's massive. All right, I got one more. Grace, go ahead.
Just real quick, when you're saying tissue samples, like what does that mean?
It's a little bit of an ear, it's a 2 millimeter punch, basically like a small sterile hole punch that we do from the other ear. So I tag one ear and then I take a small hole punch out of the other ear. And we don't do it in the center of the ear or anything like that. It's almost like an ear piercing on a human. We do right on the corner, and then, you know, we recapture the individual. We'll kind of follow that space around so that we're not affecting the mouse's ability to hear or anything like that. It's just trying to get the most minimally invasive tissue sample we can. Some folks do tail clips is really popular in the research, but we do not like doing that because the tails are then dragging through the dirt and it's kind of an open wound. So we are trying to do whatever is the you know, best case for the mice. And we hope that after we're done with it, the mice are better off. They got free anesthesia and peanut butter, but we pulled ticks off of them. So hopefully they're a little healthier at the end of the day. And I think we are at time.
Good.
Perfect.
Thank you very much.
Thank you.